we are here in biology of the brain, how the brain is developed. And I'm doing free fertilization, how the egg meet the sperm and the process it goes, and the polar body, the first meiotic division, second meiotic division. When we have first video now, go and visit that video before you come here. You see, you do zygote formation. You know how zygote is formed. So now from zygote formation, it undergo other cell divisions. And on the area, into the first one, you the zygote. You say zygote. Right? Zygote also okay. Genetic material from the mother and father, paternal and maternal chromosomes. They are genetic material now annexed into your friend zygote. Right? But am I really saying with the zygote no, it is here where the ovum nakasa, where the barrier. So barrier when I'm saying the zona pellucida. Zona pellucida. Zona pellucida. What I say. In this house, we consider the bone could do certain stage. That's fine. And from zygote, the nucleus will divide, the cell will divide into two cell stages. And from here, the cell will divide into, I'll have, let me use this one. So I have this cell. And I have my zona pellucida around it. Into one cell. That is zygote. One cell. I say I be divided into one say those double DNA copies not forming one copy will one copy of heart. Into these two cell stages. If we start two cell stages, we are going to be divided. No better than 4 cells stage, 4 cells. Into from here, it will divide again. Still have my zona pellucida as the barrier intact. I also have my cell division. I will have 4 cell stages. Into from the zygote, one cell or one nucleus, and one cell. It will be divided into 2 cells. And as gestation period increases, it's divided into 4 cells. Into this stage, you find the blastula. The blastula. Blastula stage. Blastula. Into 4 cell stages, they say could divide into about 8 to 12 stages. Okay, from 8 to 12. Into from here, I still have my zona pellucida intact. And see, I'll be having my cells. 1. And so these are 8 to 12 stages, 12 cells division now. Near found the morula. And blastula, we have 2 cells, 4 cells. That is blastula. Before that, we have the zygote. We have the zygote. Alphabet marker. We have the zygote. We have blastula for 2 to 4 cell stage. Now I put 8 cells. Morula. Are you okay? Now, from the morula, same morula, as the gestation period increases, something strange also happens. What is happening over there? Now, from the morula, and I know this one is my morula, still my morula. From the morula, something happens. Now, I say, cells never are seen. It will be so compact at the center, maybe so accord the periphery. And this same morula, my zona pellucida still intact. And then, some of the cells are compact at the center. Maybe so lower at the periphery. And those at the center, still morula. Those at the center and those at the periphery at this stage will be a benedict. And those at the center, no, I'll call them the inner cell mass. Inner cell mass. Those outside, I find the outer cell mass. Again, from my zygote, the cell divides into two, then it divides into four. These two cell stage and this four division of cells are called the blastula. From here, now I divide the beam into 8 cell stages. My friend the morula. Morula, at the point in time, when we say cells in the bone, am very compact at the center. A being soon at the periphery. Into those higher cell center, made from the inner cell mass. Now around cells now around my friend the outer cell mass. Now, inner cell mass, out of the inner cell mass, a baby is formed. Into a forming from the inner cell mass. Now, outer cell mass may provide the material to support the growth of the baby. Into the outer cell mass, being the chorionic membrane, the embryonic membrane, the amniotic membrane, and then placenta from the outer cell mass. But the inner cell mass of the baby near the air forming. Do you get the point? Now, so there is that. So let me make it clear. Okay. And these are the center. I'm very compact. And from the inner cell mass. Now, from here, another thing too happens. At this stage, the zona pellucida starts to melt away. The zona pellucida starts to go away. Into a balance now, there will be accumulation of fluid in so in kind of side room. Into I have my zona pellucida being melt away. So into a balance now, I have my outer cell mass all right. But since the zona pellucida, that is the membrane, air go away, there will be accumulation of fluids. And so everywhere room, accumulation of fluids. Accumulation of fluids. And the inner cell mass only remember one at one side, right? And let me say I'll be having the inner cell mass here. Now hammer will be the accumulation of fluid. And the fluid about the room has until the accumulation of fluid. A banasa at this cell, my friend of the blastocyst. Blastocyst. That's saying the zona pellucida air melts away. The blastocyst. 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 So now I say the inner cell mass now has seen Messana flow because now it is different from this cell. And this inner cell mass now compact after the room. Now this stage any the same as this stage because I say there is no fluid accumulation after the zona pellucida also intact. But I say zona pellucida air go away and a fluid air bomb. Do you get the point? And I'll call this mass of cells here embryoblast. Embryo. Now I'm afraid the outer cell mass name I say into trophoblast. So, and that is the blastocyst. And I have again, let's say, with zygote formation, egg meet the sperm, go through all the things I was talking about, another form is zygote. If we zygote the cell, which is one over here, with the barrier the zona pellucida cell intact, the cell will divide again, and we will have 2 cell stage. From here to divide again, and you have 4 cell stage. This stage is what referred to as the blastula. Okay, in the sense the blastula. If you have a bit divided into 8 cell stage, now find the morula. Gestation period increases. Now, some of the cells will be much compact at the center, it means lower the periphery. From the inner cell mass. No one at the periphery and from the outer cell mass. Now, as it increases again, there will be melting, the zona pellucida will melting away. And the balance have fluid, so basically in a real room, you get a point. A balance the inner cell mass number some bone or upper parts of it. Now if you ask the accumulation of fluid. In the same logo, the morula, but as I go through differentiation until I'll still call the inner cell mass now as made from the embryoblast, because when they from the embryo, that is the baby. In some cases, the inner cell mass has and no and the air forming avalanche until another embryoblast. There are cells that are rapidly proliferating that is going to form the embryo, and at the outer ones from the trophoblast. Into the inner cell mass and now where the embryoblast, and then the outer cell mass where the trophoblast. What I say, and that is, and they do this stage from the blastocyst. Now from here, you'll be getting finger-like projections. I'm not pronouncing. Last point notes, finger-like projections. You see when I'm getting a ring, and I have my embryoblast. The inner cell mass, I see my reference on the embryoblast, right? And I'll be now trophoblast. The outer cell mass now, so from a higher accumulation of the fluid. There will be finger-like projections over here. There will be finger-like projections over here, finger-like projections. Okay, finger-like projections over here with some defined cells. From the blastocyst, there would be finger-like projections with well-defined cells, right? Well-defined cells. And I'll call them, yes, with well-defined cells. And then these finger-like projections, and then we're going to implant the endometrium part of the womb. And they're going implant as the implantation as occurs. And the implantation occurs, we go there to secrete some chemicals to melt some parts of the endometrium. Now it get sticky, it get attached, and then implantation begins. And these finger projections, right, they will called. Now we have a membrane that contains only one cytoplasm for all, and that we call the syncytial trophoblast. And then I know cytotrophoblast and then syncytial trophoblast. Cytotrophoblast. And I feel with the membrane that contains all the nuclear, all the cytoplasm for all, we call it syncytial trophoblast. So the next time from here, we will be talking about how from the inner cell mass try to form the two laminar disc, and from the two laminar discs, the third laminar disc is formed, and then we start getting our three germ layers, and then we get to go to neurulation, we go to vesiculation, we are all this formed. We say anything to me if I could do how the brain is formed. If we are in the primary vesicles, in the secondary vesicles, then we go in like that, going to know the prosencephalon, myelencephalon, all this metencephalon, myelencephalon, and all that. Now if we see the brain, then we're looking at the encephalon, we'll be looking at the nuclei, we'll be looking at cerebellum, we'll be looking at cerebrum, we look at all the components. Now if we see the central nervous system. Gradually, I will see the nervous system. But maybe now under the nervous system. Until from here, define the embryology, you will start with the nervous system. And trust me, you'll be good. Thank you.